Hello, everybody. So I'm glad to see you here. And okay, so today is a very interesting topic. We talk about plants. And actually, as you know, everything is done because of love. And I love transcription factors and their binding sites in genome and in promoters. So that's love of my life. So I know not much about plants, although my study was in the Institute of Plant Research, but still I am quite far from plant biology. That's why I invited today Dr. Maria Kozlova from Osnabrück University. She loves plants. So Maria, are you here?
Hello.
All right. So you see a lot of plants around here. And so I ask you to help me with this topic today. And especially we were thinking what kind of interesting plants to consider today, and they should have genome sequenced and should have some more information and it should be somewhat interesting. And then I thought, well, we have a coffee break with TRANSFAC, so why not talking about coffee? Yeah, you see here. I guess, Maria, you have coffee plant, right?
Yes. So why not to talk about coffee on a coffee break? Okay. So we are having a coffee break, and the reason many of you start your day with coffee it's caffeine content. First, I want to talk a little bit about the range of purine alkaloids in plants, which are, as you can imagine, a type of alkaloids derived from purine nucleotides. So there is a range of these compounds that are used by different plant lineages as insecticides. And what is interesting is that unlike many different types of alkaloids, purine alkaloids, biosynthesis involved separately in distinct lineages of plants, several of them of which are economically relevant. And plants, even in the same genus, differ in their content of different types of purine alkaloids. For example, caffeine can be found in at least 7 different plant lineages, the most well-known being different species of the coffee genus, Camellia genus, which is the tea plant, cola nut, the cocoa plant Theobroma, and also Paulinia, which is known as guarana, and also different species of ilex, such as one that is known as the mate plant. But interestingly, even in the same genus, such as in case of Camellia, not every species have similar contents of such alkaloids. For example, the Camellia ptylophila, known as cocoa tea, has naturally very low levels of caffeine, but has a high content of theobromine, which is also very prominent in cocoa. Also, in the coffee genus, the alkaloid profile is different. Talking specifically about coffee, this is a genus of paleotropical plants, normally shrubs or trees. And while it contains almost 130 different species, only 10 of them are known to have ever been attempted to be grown by humans. And many coffee species are actually not known to possess a detectable caffeine content. The 2 most relevant coffee species are Coffea arabica, which is the most widely grown coffee, and Coffea Canephora, also known as Robusta coffee.
So the one which you have in your hands is which one?
Yeah, this is the Arabica.
Oh, right. Okay.
Which is around 60 to 70% of all the coffee grown, the remaining being the Robusta. Arabica is actually a polyploid hybrid of the Robusta coffee and another species of coffee, Coffea eugenioides. Robusta coffee is the species with the highest caffeine content of all tested species, while eugenioides has lower caffeine content. So the resulting hybrid has a somewhat intermediate coffee content. Typically, espresso blends have a mix of both types of coffee. Robusta, due to its higher caffeine content, is also a more resistant plant to insect attacks or diseases, and is somewhat easier to grow, but it has a different flavor profile that many might find less pleasant. In general, what can be said about coffee is it's the second most exported commodity after oil. So one can expect a wide range of genomic and transcriptomic studies aiming at improvement of the different agriculturally relevant traits of coffee plant. But however, the pathway of central importance for us today will be the biosynthesis of caffeine in coffee. So in different types of plants, both in camellia and in coffee, caffeine is synthesized from xanthosine, and its biosynthesis involves 3 consecutive methylation reactions, which are catalyzed by N-methyltransferases. However, this xanthosine itself can be derived from 2 different pathways, either from guanine nucleotide degradation or from adenosine degradation. As you can see here also, theobromine, which was mentioned on the introduction to purine alkaloids in plants, is also an intermediate step in caffeine biosynthesis. So inhibition of the last step of this pathway would result in the accumulation of theobromine, such as happens in cocoa plants or in the cocoa tea, where the last step is not very efficient. So for the Robusta coffee, the genome, the complete genome was sequenced in 2014. And of course, the caffeine biosynthesis pathway is one of the pathways of interest for the coffee plant. And an interesting feature of the Robusta coffee genome is that there are actually multiple sets of N-methyltransferases in this organism, including the 3 proteins previously experimentally described as catalysts of 3 steps of this pathway, which are outlined in violet, orange, and purple. They also happen to have the highest expression profile in coffee plant tissues, and interestingly, both in vegetative parts and also in the developing seeds. But apart from these genes, there are also a large number of homologous proteins. All these 3 enzymes they belong to the same protein family and are quite closely related. And they form several gene clusters in the Coffea Canephora genome. Coffee has become a very socially important drink, but many people choose to have caffeine-free coffee. However, normally it is produced by chemical-based processes, which might result in quite altered taste of the resulting product, which means that it might not be as enjoyable as original coffee with expected caffeine content. So, of course, attempts of having a coffee that would be naturally devoid of caffeine were made. For example, one of the parental species of Arabica coffee, Coffea genioides, has a way lower caffeine content than Arabica, and is also reported to produce a very a drink with a very good taste profile. However, due to partially due to its low caffeine content, it's also quite difficult to grow. It has way more narrow ecological requirements than the commercially cultivated species of coffee. Therefore, the coffee produced from such a species is actually quite expensive. So one might find it for something like €200 a bag.
Wow.
So that might be the most expensive coffee that exists. There are also, for example, attempts to hybridize Arabica with other wild coffee species, for example, Coffea racemosa, which is also cultivated in the southern Africa. However, still the resulting coffee is not exactly caffeine-free. And the coffee species that are caffeine-free are difficult to grow because the caffeine is there for a reason. So it is not really clear how to solve this conundrum. There, of course, have been also attempts to perform traditional selection on Arabica itself. There was a strain identified that has a way lower caffeine content due to a natural mutation. I remind you that there are multiple paralogs of these enzymes involved in caffeine biosynthesis, which means that simply deactivating one enzyme would not lead to a complete lack of caffeine in the resulting plant. But even this strain with 0.3% caffeine in the beans is way more difficult to grow than typical Arabica. For example, farmers growing it report that 1/3 of all newly planted coffee of this variety die in the first year. So
so they need coffee to why coffee plant actually need coffee? They drink it? What do they do with it?
Coffee plant needs caffeine to be protected from all the nasty things around it.
All right, I see. Yeah.
Primarily insects, because for them it acts as a neurotoxin. For us, it also has a neurological action, let's say, but it's the insects. And while coffee is such an economically relevant crop, the biosynthesis, the caffeine biosynthesis, while the pathway is understood, even though one of the enzymes actually has never been purified, and we still do not know which gene actually catalyzes the reaction, which has been experimentally studied, but we do not know which enzyme specifically does it. But also the regulation of the caffeine biosynthesis is not that well understood. For the caffeine synthase, which is the last step in the biosynthesis, a promoter has been described, but in general, it is still somehow quite poorly understood. I believe that the answer to how to have a coffee plant that would be both robust and still produce caffeine-free beans might be in meddling with the tissue-specific expression of the caffeine biosynthesis genes, as we saw before, that they are expressed in all tissues, even though not all of the enzymes to the same degree. So we decided to look at what's going on with the availability of genomic sequences and RNA-seq data. So complete genomes were sequenced for 4 coffee species. As you might see, most of these assemblies are very recent. So there have not been follow-up omic studies for at least 2 of the species. One of the species listed, Coffea Canephora, is an endemic from Comoros that actually does not have caffeine because it does not have caffeine synthase. But point to what I noted before about coffee species that do not have caffeine. For a reason, the Robusta coffee is one of the most widespread species actually, and one of the toughest ones. So we decided to focus on the Coffea Canephora Robusta coffee genome assembly from 2014, as a number of gene expression studies were mapped onto this genome. We unfortunately could only find one RNA-seq dataset for this species, which is surprising given the economic importance of coffee species, but there are not so many datasets in gene expression omnibus, and most of them include some kind of microarray profiling. So this only RNA-seq based study actually looks at somatic embryogenesis in Robusta coffee. What is somatic embryogenesis? Well, as you can imagine from the name, using an induction by plant hormones, it is possible to promote embryo formation from vegetative tissue and use that to reproduce the plant. So it's an economically important and quite popular way to reproduce different plant strains. As coffee is a perennial plant, normal reproduction from seeds, for example, takes years, which is another problem in breeding of coffee, and also attempts at perform genetic engineering in these plants. So vegetative reproduction is the way, and somatic embryogenesis is important for industry applications, let's say. So what we decided to do is to look at their data and see what we can say about the transcription factors we could identify for the genes which expression changes on different steps of somatic embryogenesis after induction. And quite unsurprisingly,
yes, maybe before you you explained this, you know, yeah, that's something we found during the several steps of analysis. I would actually show how we did this in the software, because I guess you know, for those who are coming here, would be interesting to see.
Some
Right.
kind of action on how to do this.
Yeah, maybe
So-
we can make an interruption, and then we return back to you to see actually what does it tell us. Actually, you know, what happens. So I will share my screen if you stop for a moment the sharing your screen. So basically, as you can see, we, of course, with the idea of producing a new plant which would be growing well, but producing less caffeine, that would be an interesting task for you know, for gene engineering, or creating some transgenic plant perhaps. And of course, as you've seen in the pathway which Maria showed, it's a complex pathway, and if you block one enzyme or two, you may not get the caffeine at the end, but maybe plant will die because this enzyme is needed for early stages of development, for instance, in some other places, and that's partially why we look at this study. We are looking at this this paper not only because it was the only one available RNA-seq for on coffee, but also it's interesting actually study because there was a very early stages of development of the plant, and we would like to see whether those enzymes which are involved in coffee in production of caffeine are actually already starting expressing at this early stages of development, maybe by engineering actually regulation of those genes. We can think of re-engineering this plant in such a way that will be still growing well in the early stages, on later stages of development, but in the stage of producing the caffeine in the seeds, somehow this process will be will be stopped or will be regulated in the right way. So we have to talk about regulation, right? And to talk about and to study regulation, it's important to to analyze promoters of those genes which express these enzymes. All right, so and let's have a look at our software. So as you can see that if you have this access to our software, so you log in with your account, and then you are here in the geneXplain platform, where you can study your gene expression data, for instance, or pathway data. And but the first thing you have to have is you have to have a genome. And as you may already been using our platform, you know which genomes are there. So let me actually increase a little bit the font so you can see it more clearly. All right, so you see here the geneXplain platform. And if you're not familiar with it, you just please register for this platform. You can see many tools here which are available to analyze RNA-seq data or microarray data or many different omics data. And but today we are really focused on one type of data, namely RNA-seq, and one type of species, on plant species on coffee. And okay, and if you look here, what kind of genomes are available in the platform, you see here, of course, you know, human, mouse, rat, and other model organisms. And for, from plants, we have actually only one genome available, which is Arabidopsis thaliana, of course, and that's it. So it means, of course, if you have data on Arabidopsis, you can easily upload it in platform and do many things using not only individual tools, but also complex workflows to discover differentially expressed genes, to find networks, to analyze promoters of those genes. We have these complex pipelines in case the genome is already here inside the tool. But in the case we don't have a genome, we first have to loaded into the system, right? And that's exactly what we did. So first we had to find this genome, and there are many places to find it, various genomes. Actually, let me open it here. I had this folder. Yes, I think it's here. Right, on this site, on this site, you can find this genome, which we were talking about. And what you need is actually, you need a FASTA file. So of the whole genome. So if it could be not necessarily the complete build, so with all chromosome organized. And actually, as we announced, we wanted to first to analyze a genome from T, various types of T, but it appeared to be it's not easy because the T genome is often has a very bad state. So there are a number of contigs, but the full chromosomes are often not complete. So in case of Coffea, we were lucky to see a good build where we have, chromosomes and not very few like contigs which are outside of chromosomes. So first you load your FASTA file. And then what we of course need is actually the annotation of the genes on those genomes. And with that, you load from here this GFF file, which gives you the gene annotation. And so what exactly we did here? So just to show you again what you see when you're inside the platform. So you have here your project, which I have created for myself. You can create your projects when you have an account for the platform. And then in this project, you can, in the data folder, you simply can load the genome like that. So you, for instance, you just click here, you select the FASTA file in your before you first load it in your computer, or you can actually directly load it from the URL, just putting the URL. So you select this FASTA file, and then it recognizes, it's loading it first here. So it's a big file, and it recognizes this FASTA format, and then you are there. So I will not do it again because I did it. So basically it's here, and it has this little icon in front of it. So it means it's a genome. If you click on it like this, so it actually opens it in a kind of browser, so where you can select different chromosomes, and there are 12. Actually, there are 11 chromosomes and also 0, which I guess this accumulation of all not mapped contigs. But then when the same way you load the annotation of that of those genes. So and that's the file which is here. So if you simply drag and drop it here, so this is GFF file, and you see immediately annotation of all features from genome. So you see here the genes, the RNAs, mRNAs, and the coding regions of that mRNA. So of course, the plant genome is a regular eukaryotic genome and has exons and introns. And actually, this is a pretty big genome of Coffea, bigger than Arabidopsis, but well, still manageable. All right. So, and if you look at annotation of mRNAs, for instance, you see a lot of annotation, and that's another problem of new genomes. So the annotation is done in different ways. Different groups, they have different accession numbers, and especially if you do your research with microarrays rather than with RNA-seq, you may give also special accession numbers from microarrays. So that's complicating all your research. In this case, we were lucky that actually these genes, they have kind of standard accession numbers, which were shared also with RNA-seq data, and that makes us, easy to do the work. Okay, the first thing, but before we going into these steps, how we extract promoters of those genes, let me first, let me next also load the RNA-seq data. And RNA-seq data, which we mentioned today, coming from this gene expression omnibus entry, it's pretty recent. So as Maria said, it was actually somatic embryogenesis, when you take a little leaf and then with some actually trigger, and in fact, there is a hormone called auxin, right? Yes, yes, right. It plays a magic role in this development. And they actually, in this paper, they what they did, they have a point of induction of the development at time 0, and then they have measurements of gene expression before that time and then after that time. So 1 day after, 2 days after, 21 days after. And that's the data we want to have. So basically, we have here a link to that HTTP address of that data, and then we can again here in our folder simply upload this, put here this link in FTP, and then by OK, you will load it into the system. So again, I will not load it right away, but I will rather show the results. So you see what are the results? Actually, the same set of data which you've seen just on your screen, but here they are already in the system. So it's in a set of tables where we have this accession number of a gene and the column which tells you the count. So basically, when you do RNA-seq, you sequence RNA many times with you chop it. Well, basically, you sequence it from using either from one side or from two sides. Here, this one side sequencing, and then here they did it in twice in each time point. So, and then this number is a number of reads which you sequence from that protocol. And the higher the number, the more expressed is this gene, right? Of course, you have to normalize it and do many other things, and that's exactly what we are going to do quickly. I don't bother you with many small details of the analysis. So basically, what we get at the end is one table where you put all these counts inside one table. I think that's the one. All right. So here you got such a table where you have your accession number in the first column and those counts of the reads in each column corresponding to this time point. All right. So, and then to detect differentially expressed genes, we have a number of various methods. And one method I love is this methods called, guided Limma. So here in the method section in our platform, in statistical analysis, and there's a number of statistical methods. And one which is very powerful is called guided Limma linear model analysis. Or guided Limma, which is a pretty powerful tool to detect your differentially expressed genes. So what you have to do is actually you have to prepare a table where you describe your samples. So a table like that. So where you're saying that in this data table you have those kind of columns, and they correspond to those time points. So, and you can have many other columns corresponding to other other factors of your experiment. If, for instance, if you did experiment into batches, you can put here the batch number or something like that. Those kind of factors can be involved also in the detection of differentially expressed genes. In this case, this was done all in one batch, so you don't need to add any other factors. So what then you have to do, you take this table here, So table with your data, and you take the sample table here, so which tells you which sample belong to which category. And then you have to select normalization method, which is quantile, for instance. You select those columns you want to analyze. And then in this table with the samples, you are saying which column is correspond to the sample column, and which column on that table correspond to the main factor. So basically, the main factor is the feature I want to use to compare things to each other. And then you have to give the reference level, which is 0 dBi. So 0 dBi is my reference level. And then you do comparison to reference only, which means you can compare many different factors to each other as well and get all kind of comparison also between different time points. But if you want to compare to the reference point, to the time 0, you simply select it here. And here you can select other factors which you want to use as a control. So in your normalization procedure, in this case, we don't have any other factors. As I said, in case if you have different batches, for instance, you can put them here. And there are other tricks like with SVA or to make it robust. So you can read about this method. There is a number of papers describing it. So then your run is ready and simply you start run. So it takes a bit of time to analyze this data, but at the end, what you get is this kind of tables, which tells you, each comparison which we did. For instance, here we did compare 14 days before induction compared to the day 0. And here, for instance, 1 day after induction compared to day 0. And then here you see those genes which are downregulated. So the log-fold change is minus and significant. So even FDR, so which means that p-value is which is adjusted to the multiple testing is corrected. And then you have also the upregulated genes. And as you can see, there are quite a number of up and down-regulated genes. And with that, you can do it for each comparison. What I did, I simply combine all them. But actually, one thing which was even done better, so I have it here in the Limma results. You can see it in one table, which is called ANOVA results. So where you have all these various types of comparison all together, but then you have one p-value, which tells you whether in any of those comparisons you get the significant change. Well, in a simple wording, it's kind of ANOVA p-value, which you can use to select those genes of your interest, right? And what I did, I selected these 1,200 genes which have this p-value, adjusted p-value of ANOVA, actually below 0.02, so such a cutoff which gives you a significant differentially expressed genes, but still it's so sort of soft cutoff which gives you enough genes to look at, right? Okay, so we have this 1,200 a bit more genes. And now we want to play with them. What do you mean play? So, for instance, we want to understand which of these genes are expressed in which stage of development. And maybe we can find such a cluster of genes, which has in fact developed in the same way during different steps of development. So you mean, what I mean, you need to cluster this table in a various ways. So first, what I did, I actually made a kind of a heat map of that cluster. You can see it here. So where what we see here are actually those five conditions. So here we see 14 days before induction, 9 days before induction, 1 day after induction, 2 days, and 1 day after induction. And as you clearly see, actually, on that picture, very important time is of just after induction, first day of induction. And you see a very huge number of upregulated genes and downregulated genes, which has just happened in this one day. So this is our special focus. Anyway, basically, then what I did K-mean clustering of that set of genes. And for that, again, just let me show you among these statistical methods, we have this K-mean clustering where you can put your table as it is, and for instance, like this, and then you select, let me put it like this proper, and then you select which columns you want to use for clustering. Then you select which method of clustering you are doing, and then you say how many clusters you want to see. So I put here, and I remember, I guess, 10 clusters, and then I got this these results of K-mean clustering where each gene is if reflecting in the cluster. So you see we have here 6 clusters actually, and the one cluster number 1 is have a quite number of genes. What we see here, we just selected this cluster, and we get 157 genes with cluster 1. And what is a cluster 1? What is important for cluster 1? You can see this is actually it's characterized by high expression in the first day after induction, and also pretty high expression of the second day after induction. But in the first day of induction, this cluster 1 was important. So all these 150 genes are, you know, they have, most of them actually, they get boosted in the first day after induction. All right, so we have this set of genes. We can actually annotate them by adding annotation to those tables. And to do that, again, we have this very good method called annotate table. It's in a different location here among the data manipulation methods. So you can add to any table of your interest information from any other table. And for that, you need just the same accession numbers. We have even a super annotate method here. It's also very interesting when you can annotate not just using the accession numbers in the first column, but using any column in your table to add annotation to that column. So we have this super annotate method as well. But here we just use this simple annotate table method where we have now our 157 genes. And we know what these genes are doing, you know. And what is important, actually, we have the position of those genes in genome, right? Why do we need it? Because we need promoters of the genes, right? So, and promoters are in the genome. So we need to extract from genome those sequences and get their sequences, right? So for that, we have, first we have to convert this table into a track. And there's a special method, which is called convert table to track. And then, to make this story short, so what we did here, we have now this track with all the genes which have upregulated, well, which in the first day of induction, and they belong to this cluster of genes. We have similar behavior in this process of the very first steps of development. So that's important gene, and they may be controlled by certain subset of transcription factors. Which ones? That's we have to find out. So we have the gene, but now what we need actually the promoter of that gene. So we need a track which is upfront of each of that gene. So to do that, we have various types of methods here to convert. Actually, what we are doing here, we work with this set of tracks, and we want to add manipulate with that to create a little distance to it. So that's exactly what we did. That's another track. As you see, this is a track of promoters of those genes. So we extended this track a little bit, 1,000 nucleotide upstream of start of transcription and 100 nucleotide downstream of it. Okay, so we have this set of promoters. So what else do we need? We also need a set of promoters which we want to compare with. And we want to compare with promoters of genes which didn't change expression over all this period of time. They just still come, you know, they are not changing their expression. So we get a set of such promoters from the same experiment. So now we have these promoters here. So these are promoters of genes which didn't change expression. So now we have promoters of genes which did change expression and promoters of genes which didn't change expression. All right, so next, what we need to do, we need to find transcription factor binding sites in those promoters. And to do that, we have here among the methods of site analysis, we have this method of site search on track. So actually, what you do here, you put this track like promoters here, and it automatically selects which genome it is because this track is connected to that promoter track. And what is important is which set of positional weight matrices we have to select. And that's something we have to make a little stop here and look at TRANSFAC database as it is. So as you know, maybe TRANSFAC database is one of the biggest collection, is actually a biggest collection of positional weight matrices from various organisms. We have collected more than 10,000 matrices. And for plants, we have a special collection of matrices here, which have 1,500 positional weight matrices, mainly coming from Arabidopsis, but not only. And here it's a point. So of course, the coffee plant is not well studied, so we don't know really many transcription factors in this plant. So we have to use motifs, positional weight matrices, from transcription factors of Arabidopsis most of the time. And why we can do that? Well, because we are brave, but also because actually we can assume that DNA binding domain of many factors is not really changing too much. It's there, we get families of new factors in different species, but the closer the species in one to each other, the less variability we can expect. So it's a big guess anyway, but that's what we have. So that's how we do. We simply put these plant PVMs here in this program, and we run it. So, and interesting to see that although this matrix is coming from Arabidopsis, but indeed you find a lot of matches of them in the promoters of the coffee genes. So here, what you see here are the sites which are predicted for each of that PVM from that plant library. Of course, you see there are so many sites found here, and it would be interesting to find out which ones are more important, which ones are less important, and how to find it. So for that, we have these algorithms of searching for enrichment. So, and for that exactly, we need these non-change promoters. So we compare which sites are found here in the promoters of the changed genes, and we then see which sites are found in the promoters of non-changed genes, and we compare those two things, and then we get such output like this, where you can say we found 176 PVMs, basically motifs, which are enriched in promoters of these genes of our interest compared to promoters of non-changed genes. And it calculates this enrichment factor, which tells, for instance, for the first factor, which is FUS3, it's more than seven times higher enrichment in promoters of these target genes compared to non-change genes, and there is a significance in it. Still, we have quite a lot of factors here, and many of them, they might find the same motives. So it's very difficult to understand which factors plays a major role in our development. And for that, we have a second algorithm which searches for composition of factors, and the results of that, what you see here. So it found actually this composition of motifs, which you see here, which discriminates the promoters of upregulated genes compared to the promoters of non-change genes. And you see this set of factors now much smaller. All right, that's how it works. But let me briefly come back to this coffee production. So when we look about coffee production, what have it to do with this plant development? Maybe nothing. Let's have a look first as a coffee pathway as it is. So actually what we did is we took this coffee pathway as a set of genes. It's actually, this is caffeine pathway. So it has 23 genes which are involved in the production of enzymes, or maybe these are including also paralogs. We don't know in which exactly they contribute, but we put them all together. And again, the same way we extract promoters of those genes and so forth. And when we run it here, let me see, no, that's this run. The final run is this one. So finally, we found now this set of transcription factor motifs, which discriminates our sets of genes. I mean, the sets of these genes which are coming from this pathway from the background set of genes. So, and this discrimination is very good. So actually, although the set of genes is pretty small, but discrimination we achieve is very high. And now we can have a look at these transcription factors and see whether they really play an interesting role in some development or maybe in caffeine. And now with that, I want to give back to Maria so she can explain me. So for me, it's just, just some motifs, but for her, it's, I guess, it's full of meaning, all these transcription factors, which we found. Can you show this to us?
Yes. So looking specifically at the caffeine pathway. So of course, it's not so easy to stretch the annotation of Arabidopsis transcription factors to coffee.
Right.
But for this set of transcription factors, I also searched for the homologs in coffee, and I could find quite closely related genes, even for the transcription factor, which originates actually from rice and not Arabidopsis, like the last in the list. So it's not so easy to deconvolute what is going on, also because not all genes have a good description of what exactly they do even in Arabidopsis. But what attracts my attention is that there is a gene known to be a floral repressor, which site is found in one of the modules. And there is also let's say general regulator, one of the homeobox proteins.
Mm-hmm.
And there is a protein involved in breaking seed dormancy, which means that the caffeine biosynthesis would be activated a germinating seed,
All right.
which makes sense. It was shown that actually young seedlings have quite a high caffeine content, and then the plant keeps having it in vegetative tissues. So it's because it's also very important to protect the very young seedling. It doesn't have any mechanical ways of protection. So the only way it has to protect itself are these compounds that it synthesizes. Some of the other regulators were described to be involved in resistance to specific types of bacterial infections. It is not so clear for me how to relate this to coffee. And also there is a site for a protein that was described to be involved in salinity tolerance in rice. And in general, the factors listed here are described to be involved in some kind of stress resistance in the studied plants. So I'm not so sure how the fluororepressor relates to this, because caffeine synthase are expressed in, as I was showing before, in all tissues. They're also expressed in flowers. Actually, interesting thing about flowers that caffeine is present in the flowers of citric plants, but only in flowers. For citrus, it's not in leaves, in foliage, it's not in fruits, but only in the flowers. But for coffee, it's everywhere, even in the roots. When we were looking at the expression of different paralogs of the enzymes involved in the caffeine biosynthesis pathway, apart from the main 4 enzymes, the other ones, they're not so universally expressed, but they also way less expressed. So maybe this is also something that we should look into to be able to see if caffeine expression can be modulated in some external way in this plant. But I could not find much difference between different NMT genes in terms of who regulates them. They are quite uniform in this terms. And of course, we can take this with some grain of salt only because, of course, there are very small number of transcription factors described for coffee. But, I think that still the fact that we find some transcription factors and somehow come up with some meaningful relation of them to the development of coffee plants is actually interesting. When we looked not specifically at the caffeine pathway, but in to the general changes in expression, I believe that it is interesting that there is a number of transcription factors that respond to different, are involved into response to different plant hormones, such as auxin. So that also somehow validates our approach of using the matrices from Arabidopsis, as the paper itself describes the auxin-mediated regulation. There is also like another hormone that I've seen reappearing among these transcription factors is the abscisic acid. So as our analysis is quite different from what was attempted in the paper, but we still see the expected auxin response factor here, it also shows that using this approach for different plant species is actually a worthwhile approach. So, of course, people working specifically with plant biology might be able to extract more information from the transcription factors one would get in such an analysis. I'm not a plant biologist myself, but still it is quite interesting that we can see first see the similar regulators as described in the paper that uses a different way to approach the regulation in this plant, and that we could see how on the early stages of plant development, the economically relevant and relevant for us caffeine biosynthesis is also regulated. I looked at the tissue specificity for these genes. For I'm not sure, maybe I can show it. For Arabidopsis, there is a way to check the tissue specificity of like expression of different genes. Okay, do you
yes, we can see it, right? Yeah.
So, okay, so we put it like that so we can see the gene list. So for every type of, for every gene of Arabidopsis, it is possible to see the annotation and to see where it is expressed. So, for example, this factor is quite strongly expressed in almost every kind of tissue. So we don't really see it being differentially expressed during seed development or seed germination. Like here's some slight increase. And I checked different genes we found here, trying to see if there are any that are expressed only in seed development process, because that would be interesting. So we could think of the ways to modulate the expression only in seed development and not the general plant life cycle. As I said, that it confers quite a good resistance to the plants. But I couldn't see anything that would be specifically differentially expressed only in seeds. But I remind you that this expression data is still for Arabidopsis, so maybe the same factors might be expressed quite differently in the coffee plant. Exactly. I see that there are some very new omics studies for the species from this year, that the data cannot be just retrieved from Geo at that point. But as there is an increase in number of them, and there are also newly resolved and resequenced genomes for other coffee species, I think that maybe we will be able to come closer to answering these questions soon, as there is finally more data on coffee. And but still, I think it's a nice example on how much information we can get on even on a plant species that is not that well studied.
Exactly. Yeah, that's right. Thanks. With this optimism that with more data we can come up perhaps with a better understanding. Just at the end, you know, like a very simple perspective. If you let me share my screen again, I actually want to you know, compare. I guess you see it. I want to compare the question. You know, we consider the basically the production of caffeine on one side, and we compared and we analyzed the very early stages of development of the plant on another side, and it looks like a different stages and different processes. But I just simply wanted to compare it. So what I did, I took these 23 genes, right? So which are producing the enzymes of caffeine production, and using these mapping tools in our platform, I just add the expression values of those genes in the stage of development, of early stages of development of the plant. And some of them, for instance, if you look at this day one, which was very important for us, for instance, this gene we had very high peak of expression in the early stages of development in the first day of after induction. And what is this? Is actually enzyme monomethylxanthine methyltransferase, which is actually has these sites in the promoter. So sites for these 4 factors. And it seems this enzyme, which is, I guess, if I'm wrong, if not wrong, it's one of the first stages of that pathway, right? So it might be quite rate limiting, is it?
It's no, I think that it's the MXMT. This is the one step before the last, actually.
All
but
right,
this
so
is able to catalyze also the last step. It's quite interesting that they because they're all very close, these proteins, they are partly able to perform each other functions. But this is the this is one step before the last.
all right,
and this
so
is
yeah.
this is the main enzyme, I would say, the one of the 4 highly expressed ones, this one.
So, okay, and we see it's starting to express in various stages of development. So it means if we really want to tackle this mechanism by gene, editing, we probably have to be careful with this gene, because if we break it, and it will not play its important role in the early stages of development, maybe the plant will not develop. So, my dream is actually, if you look now at the promoters of such genes, of such important genes, you can think about a very specific gene engineering when you're not really removing the whole gene, or you're changing the protein in some sense, so this changes a lot the function of that protein, or independently where it works. But really, you would be probably able in the future to tackle little pieces of information in the promoters of those genes to change a little bit the expression pattern of those genes. So the gene is fine, gene is there, it's expressed, but it's not expressing a little bit in a different situation, in different cell type after our editing, and that's why we may edit the plant in a very gentle way, but still in a way how we want to have it. All right, so we had today quite a long discussion, well, not between us two. We still have a little time for your questions, if you have one, a few, so we can try to answer them together. So if you have questions, please put them in Q&A.
I see a question about the coffee species we were talking about.
All right, so maybe you can answer that.
How far is your coffee species different from Coffea arabica? Coffea Canephora, which is the Robusta coffee that we were analyzing, is one of the parent species of Coffea arabica, which is a polyploid hybrid of Robusta coffee and Coffea genioides, which is another coffee species. So it is different in terms of Arabica coffee having also a gene set from the other parent. And the reason there are more studies on Robusta genomics is simply because the genome is smaller, so it's a bit easier to work with it, and that's why the assembly was first done for Busta coffee. But Arabica, it has all the same genes plus also the genes from Eugenioides. Actually, it was one of the ways to prove the ancestry of Arabica was a recent study where both parent species had their genomes sequenced again by a large consortium, I think involving Nestle and one of the other big coffee producing companies. I forgot the name. But yes, so the Arabica genome is derivative from these two species. So these genes can also be found in Arabica.
All right, so I think with that, I would like to finish, and thank you, Maria, for your contribution and lovely background with all your nice plants which you have. Today we were talking about this very nice plant, which is coffee, which gives us a lot of nice moments when you take your cup of coffee and you enjoy life. And that's what I like you to do. Thank you very much. Okay, bye-bye.